Hello guys, so today we will going to discuss about bacterial resistance, or more specifically saying antibiotic resistant, the resistance of bacteria against antibiotic. Okay, so let's discuss about something interesting. So we all know that bacteria is ubiquitous in nature, and you can find bacteria in each and every part of this universe, right? So now, although most of the bacteria are harmless, but there are some 4% of bacteria that are harmful and causing most of the disease in us, right? That means the type of the disease or the number of the disease that of which we are affected, usually most of them are caused by bacteria, right? So under such circumstances, it becomes very important to treat those bacterial infections, right? So under such circumstances, in 1940, the antibiotic was developed, okay, which is a simple and effective treatment for many bacterial infection. So now the fact is, what is antibiotic? So antibiotic is a secondary metabolite that bacteria used to produce in its stationary phase mainly. Okay, so now what is the utility of this antibiotic in terms of bacteria? Let us first understand this. So let's talk about soil. So soil, we all know, is a very diverse ecosystem, right? It consists of many bacteria, right? So under such circumstances, when let's say there is a deficiency of nutrients, that means there is a competition environment. Under such circumstances, it is important for the bacteria to ensure its survival. Okay, so under such circumstances, bacteria sometimes converts themselves into spores, which is a metabolically inactive form, and in this spore form they can live for many years, but in some cases they start secreting antibiotics. So what is the utility of these antibiotics? These antibiotics will destroy, or in some cases will inactive or will inactivate the metabolism of other bacteria. Okay, that means the antibiotics that the bacteria secreting will either inactivate the metabolism of other bacteria or causes the destruction of that bacteria, and as a result it become helpful for the bacteria for its own survival, because the competition gets lowered, right? So that is the concept why antibiotic is produced by bacteria. Okay, so it is clear, antibiotic is nothing of chemical or synthetic type, right? It is purely produced by bacteria and we are using, because after discovering about antibiotic, scientists thought that as it have the ability to inactivate the metabolism of bacteria, and also some of the bacteria causes destruction, some of the antibiotic causes destruction of the bacteria. That means antibiotic has both bacteriostatic and bactericidal activity. Okay, so under such circumstances, scientists thought that we can use this antibiotic to treat those bacterial infection that is a major problem in the medical world, right? So they start using antibiotic. Although Alexander Fleming, who was the discoverer of penicillin, we have talked about penicillin during the cell wall antibiotic class, warned us to not use antibiotic too much, or he warned us about the overuse of antibiotic, because he predicted that if we use antibiotic at too much extent, then there is a chance that bacteria can develop resistance to antibiotic. So unfortunately, nowadays his prediction become a major problem in today's world, right? Because nowadays most of the bacteria acquired resistant to most of the major types of antibiotics. Okay, and as a result, despite using antibiotic, despite using proper medication, the patient remain infectious because bacteria have developed the resistance, and as a result bacteria are now no longer be affected by the antibiotic. Okay, so first of all, let's see the role of the antibiotic in protecting us from bacterial infection. So antibiotic decrease the mortality rate associated with serious cause of pneumonia from 90 to 10%. And given this stunning result, many prominent medical community, including US Surgeon General, thought that antibiotic would effectively make bacterial disease a thing of the past. But during this time, Alexander Fleming warned us. Okay, and nowadays it is true that bacterial resistance become a dangerous problem for us. So now let's enter in details. So let's discuss this flowchart. So suppose you have suppose I have infected with a bacterial disease. Okay, so this is a harmful bacteria. So I have consulted a doctor. He understands the symptoms and prescribe me antibiotic. Okay, and for first recovery I have started to consume antibiotic in over amount. Okay, or let's say, as now I am feeling well, I have stopped my course. Okay, so in normal cases, if I use the antibiotic as prescribed by the doctor, then this pathway will happen, that it is causing the disease. So I have consumed the antibiotic, and as a result bacteria eliminated, and now I am recovering, right? But if I use those antibiotics in over amount or in much amount, and if I stop my treatment as I am feeling well, as a result what will happen, some of the bacteria, due to evolutionary pressure, acquire some adaptation. Okay, this adaptation is nothing but accumulation of some gene, and these genes are the drug resistant genes. Okay, because that means, suppose my disease is due to 100 bacteria. Okay, so now the idea is, among 100 bacteria 95 will die but 5 will survive. Okay, and because they have acquired the antibiotic resistance. Okay, so now they will develop and they will start replicate, right? And as a result a new population of bacterial resistant will produce. Okay, and as a result I will not recover despite using antibiotic. So that is the idea. That means if you use antibiotic in too much amount, or you have stopped your course just because you are feeling well, under such circumstances antibiotic resistance can develop, and this is because of evolutionary pressure. Because remember, there is a term, survival of the fittest, right? So bacteria also try to survive under such harsh condition. Okay, so it can acquire some resistant gene by two means. I will going to discuss about it. Those genes will acting as a drug resistant genes, and as a result they have developed drug resistance, and that is the concept of the bacterial antibiotic resistance. Okay, so antibiotic resistance can be of two types. There can be molecular approach or evolutionary approach. Okay, so in both the cases overuse of drug is responsible. So in molecular approach there can be chance of random mutation or spontaneous mutation. Okay, and which ultimately facilitates transcription of the new protein. Because remember, suppose there is a gene ATGCGAT. Suppose there is a DNA sequence, okay, not gene. So now let's say for spontaneous mutation the C will change into G. As a result, the GGG will code a different amino acid. For example, let's say, and due to this, the incorporation of this amino acid, there is a formation of new protein. Okay, and this new protein causes some structural and functional changes or morphological changes, and as a result, antibiotic cannot work. Okay, suppose let's say a DNA RNA synthesis inhibitor which is targeting a special type of enzyme involved in DNA replication. Okay, so now due to the development of this protein, it facilitated a morphological or functional change that changes the binding site in the enzyme, and as a result the antibiotic cannot bind with that enzyme, so it cannot prevent the activity of the enzyme, and as a result the antibiotic become inactive or ineffective. Okay, so that is the idea of molecular approach. That means due to over usage of drug, there can be chance of random or spontaneous mutation. Okay, and this random or spontaneous mutation causes formation of new protein which changes the morphological and functional structure. Okay, and that established the antibiotic resistance. Okay, and there can be also evolutionary approach. Okay, that means we are using antibiotic in too much amount. Under such circumstances there will be evolutionary pressure. Okay, and bacteria try to ensure its survival. Under such circumstances bacteria will acquire some gene that will allow them to be naturally selected. Okay, and as a result antibiotic resistant bacteria formed. Okay, and this kind of natural selection that I have told you, that among 100, 95 will die, 5 will survive. So this kind of selection is the simplest and cruelest form of Darwin theory of natural selection. Okay, so that is the idea. So antibiotic resistance can be of two types: molecular approach and evolutionary approach. Okay, in molecular approach, due to overuse of antibiotic, there can be chance of random or spontaneous mutation. Okay, and this random and spontaneous mutation causes production of new protein, and this new protein changes the morphological and functional structure of the bacteria. Okay, and as a result it develops antibiotic resistance. Okay, and another important thing is the evolutionary approach, where there is a overuse of antibiotic, and this overuse of antibiotic creates a harsh evolutionary environment or competitive evolutionary environment. Under such condition bacteria will acquire some adaptation, which adaptation is nothing but drug resistance gene that causes the natural selection of some of the bacteria. Okay, and as a result now those bacteria who have successfully survived will facilitate its replication. Okay, and as a result a new population of antibiotic resistant bacteria develops. Okay, so if we don't talk about antibiotic, you know what, some bacteria are naturally selected against some kind of antibiotics. Okay, that means some bacteria have natural resistance against some kind of antibiotic. If you have watched the video where I have told you about the cell wall synthesis inhibitor, then I think you know that penicillin targets the peptidoglycan layer, right? There are various kind of protein that is activated by penicillin that causes the disruption of the peptidoglycan layer. Okay, now if we going to divide bacteria broadly, so we will end up with two things. One is gram positive bacteria, another one is gram negative bacteria. Okay, and gram positive bacteria consist of thick peptidoglycan layer, I think you know, and gram negative bacteria consist of thin peptidoglycan layer. Okay, and above this thin peptidoglycan layer there is also a outer membrane, right? So what do you think, what will be very easy for penicillin to show its activity? Either it is gram positive or gram negative. Obviously gram positive, because it has a thick peptidoglycan layer and which is its outer layer, right? that means above peptidoglycan there is nothing, right? But in case of gram negative bacteria, the thin peptidoglycan layer is sandwiched between two membranes. One is outer membrane, another one is inner membrane, and as a result penicillin find it difficult to show its activity against gram negative bacteria. Okay, that means gram negative bacteria we can say are naturally resistant to penicillin. Okay, so now it is time to understand about how bacteria acquire those kind of resistance. So let me tell you that yes, I think you know that bacteria have circular DNA, right, which is its genetic material, right? So along with this circular DNA, it have a extra circular DNA which is known as plasmid, right? So now there can be a chance that this plasmid contains antibiotic resistant gene. Okay, and this is the most of the cases what is occurring. Okay, so as a result, when plasmid will transcribe and translate, it will lead to the formation of the protein. So we will going to learn about what type of function this protein shows. But before that, it is necessary to understand how this plasmid is acquired by this bacteria. Okay, so there can be two processes. One is vertical gene transfer, another one is horizontal gene transfer. Okay, so whenever we are talking about vertical, we think something like this, right? This is a vertical line, and horizontal line means this, right? So vertical gene transfer is the transfer of the gene due to bacterial replication. Okay, that means from the father and mother bacteria, the gene is transferred to their progenies. Okay, so that is the vertical gene transfer. And horizontal gene transfer means when the gene is transferred from one bacteria to another bacteria, and those two bacteria are of same generation. That means there is no relation of there is no relation of parents and progenies in between these two bacteria. Okay, so there are three important transfer mechanism by which horizontal gene transfer occurs. One is conjugation. So let's talk about conjugation. So conjugation, we can say it is kind of bacterial sex. Okay, because there will be a formation of pili. This pili will attach one bacteria with another bacteria, and then the plasmid that these bacteria have will be transferred to this one. Okay, although this plasmid will be transferred in the form of single stranded DNA. Okay, and then it will replicate here also, and as a result in both the cases there will be two circular double stranded plasmid. Okay, and now let's think about this, that this one, let's say this bacteria is number 1 bacteria and this is 2. Okay, let's say this number 1 bacterial plasmid contains antibiotic resistant gene. So as a result, as the plasmid is transferred to the number 2 bacteria, so there can be a chance that number 2 bacteria have acquired the antibiotic resistant gene, right? So that is how via conjugation antibiotic resistant gene is transferred to one bacteria from another one. Okay, now it is important to talk about transformation. Transformation means, let's say there is a bacteria which contains antibiotic resistant gene. Maybe it is in its plasmid or in its normal chromosome. Okay, but most of the cases bacterial resistant gene will be in their plasmid. Okay, now with time the bacteria will lyse. Okay, and as a result all the genetic material and all the important component will be outside the bacteria. Okay, because as bacteria is lysed. So now along with that, it is obvious that the resistant gene will also come outside. Okay, now this resistant gene can be acquired by this bacteria. Okay, via a process called transformation, where the resistant gene will first attach with a particular type of receptor, or let's say the cell surface of this bacteria, and then it will be acquired. Okay, by the number 2 bacteria. So that is the process called transformation, right, where bacteria is acquiring the extracellular DNA. Now the third one is transduction. Okay, so transduction is facilitated by phages, so bacteriophages. So bacteriophages are kind of bacteria, the kind of viruses that infect bacteria. Okay, there are two kind of bacteriophages, one is temperate phage, another one is lytic phage. Okay, so now the idea is the phage will inject its DNA. Okay, so now if it is a temperate phage, so the phage DNA will first integrate with the bacterial genome, leads to the formation of prophage, and when there is a appropriate condition for lysis, the phage genome will be removed from the bacterial chromosome and now it starts replicating. Okay, remember a very important concept, that temperate phage have the ability to perform both lytic cycle and lysogenic cycle. So the prophage formation that I have told you just now is the lysogenic cycle. Okay, and when bacteria and when there is a appropriate condition which is known as induction, the phage genome will be removed from the bacterial chromosome and now this phage genome will start its replication. Okay, and then it will destroy the bacterial genome. Okay, and uses the host cell machinery for their own replication. Okay, so and now the viral DNA will replicate, then production of the protein will occur, it will produce the head, tail and another base plate and another important thing, then it will assemble, and after that there will be packaging of the DNA into its head. Okay, so there can be a chance that as these destroyed or damaged chromosomes, or let's say these broken nucleotides are also present in the cytoplasm, there can be a chance that some virus accidentally package antibiotic resistant gene along with its own genome. Okay, so now when those antibiotic resistant gene containing phage will infect another bacteria, so it is obvious that the bacteria that the phage will transfer the antibiotic resistant gene. Okay, so that is how the three process ensures the antibiotic resistance of the bacteria. Okay, and what is the example of vertical gene transfer? So once the bacteria have acquired resistant gene, so now it is obvious that it will replicate and divide. Okay, and leads to the formation of two more bacteria, and then each of those bacteria will acquire the resistant gene. Okay, so this type of transmission is known as vertical transmission. Okay, so now then the antibiotic resistant gene will transcribe, translate and leads to the formation of protein. So now it is important to understand what kind of protein antibiotic resistant gene can produce. So one is antibiotic degrading enzyme. Okay, so we have already talked about this in our cell wall inhibitor antibiotic video, where we have discussed that their gram negative bacteria often develop a special type of enzyme called beta lactamase, right? This beta lactamase have the ability to degrade the beta lactam ring of the penicillin. Okay, so this is the type of the antibiotic degrading enzyme. Okay, so antibiotic degrading enzyme example is beta lactamase. So example of the bacteria that can acquire such kind of degrading enzyme are E. coli. Okay, so now let's move to the next one, that is production of the efflux pump. So due to the antibiotic resistant gene, there can be a chance that the bacteria can develop a type of efflux pump. So what is the utility of this efflux pump? So the utility of efflux pump is mostly observed in case of tetracycline, which is a protein synthesis inhibitor. So when we use tetracycline will enter inside the bacteria and bacteria will pump out this tetracycline with the help of this efflux pump, and as a result tetracycline cannot show its activity. Okay, so that is the idea of efflux pump. So efflux pump will pump out the antibiotics from the bacterial body. So another important thing is modified antibiotic binding target. So we have already talked about modified antibiotic binding target. That is, I have told you during when I have explained the mechanism of penicillin, how penicillin works. So I have told you there that penicillin will bind with the penicillin binding protein. So antibiotic resistant gene also develops some important protein that will modify those penicillin binding protein. Okay, and as a result penicillin cannot bind with those penicillin binding protein and cannot show its activity. So that's how antibiotic resistant gene ensures bacterial resistance. Okay, one is antibiotic degrading enzyme, second one is the development of efflux pump, and the third one is modified antibiotic binding target. Okay, the third type of thing you may find in case of the MRSA, that is methicillin resistant staphylococcal aureus. Okay, so that is the all about bacterial resistance. This is the first video for antibiotic resistance of the bacteria. So I hope this video will be helpful to you to understand the basic concept of antibiotic resistance that bacteria have developed. So we will again come back with another topic of antibiotic resistance. So till then, bye bye.